Hello friends, how are you? I hope you are good. In the last video lecture, we have seen the structure of protein, that is primary structure, and the secondary structure of protein. Today we are going to deal with the tertiary structure of the protein and quaternary structure of the protein. Actually, we are seeing the chemistry of protein, under which we are seeing the 4 different levels of protein, or we can say that the 4 different organization of proteins. So among these 4, in the last video lecture we have seen the primary structure of the protein and the secondary structure of the protein. Today we are going to discuss about the tertiary structure and quaternary structure. The tertiary structure and quaternary structure. But you must know that we have seen the primary structure of the protein. The primary structure of the protein is the sequence of amino acids linked with the peptide bond, and these amino acids are just in a sequence manner. And we have also seen the structure of insulin, which was given by Frederick and Sanger in the year 1953, in which we have seen the disulfide bond. The disulfide bond formed between cysteine and cysteine. It is attached with the SH group of the cysteine and cysteine. So you must know that the secondary structure of the protein, in which we have seen the alpha helical structure and the beta sheeted structure, in which 2 types of beta sheeted structures we have seen, that parallel and anti-parallel. Parallel structures means the N terminal will be ended with the C terminal, and the second polypeptide also N terminal is ended with the C terminal in a parallel, and in anti-parallel the N terminal will ended with the C terminal, and the C terminal in another polypeptide will be ended with the N terminal. So in this manner we have seen the parallel beta sheeted plate and anti-parallel beta sheeted plate. Today we are going to see the tertiary structure. In this, I am making the lecture very easy for you, and this is exam oriented answer. If a question is asked in the exam, write short note on tertiary and quaternary structure of the protein. So you have to write the answer like this. You have to start with this. First we'll see the tertiary structure. In this tertiary structure, the secondary structure belongs to alpha helix and beta pleated sheets. So this structure, secondary structure, will folded again, or the folding of secondary structure will form the tertiary structure into the 3-dimensional structure. Means that the secondary structure will be folded into 3-dimensional structure known as tertiary structure of the protein. So you can say that the interrelation of the twisted form of secondary structure will leads to the tertiary structure, three-dimensional tertiary structure. So here the tertiary structure of the protein is explained with the help of the diagram here. In the first diagram of the tertiary structure, the schematic structure of tertiary protein means that this will be the schematic structure in which the alpha helical structure is shown here and beta pleated sheet structure is also there. So this tertiary structure is the combination of alpha helix and beta pleated sheet. This secondary structure is folded into tertiary structure and it forms the three-dimensional structure. So this tertiary structure in the schematic structure, we'll see this tertiary structure of the protein will contain hydrophobic amino acids. It will contain hydrophilic amino acids, and it will also contain acidic amino acids, basic amino acid, so the tertiary structure of the protein will contain hydrophilic amino acid, hydrophobic amino acid, acidic amino acid, and the basic amino acids. So you must know that the structure of the protein in tertiary form will contain these types of amino acids. Now here we'll see the tertiary structure of myoglobin. What is myoglobin? Myoglobin is a protein belongs to muscle. Now we'll see the structure of myoglobin. Here this is a tertiary structure of myoglobin. So the structure of this myoglobin is given. You will feel it as an complicated one, but it is not a complicated one. In the examination, how to draw the figure I will show you, don't worry. So here we have seen the schematic structure of tertiary protein here. So this is the tertiary schematic diagram, alpha helical and beta pleated. Now we'll see the tertiary structure of myoglobin here. Here we have to just see the polypeptides, they are further folded. The secondary structure of the polypeptide will be further folded into tertiary structure. Now here the simplification of diagram is this. Don't worry about how to draw this. First you have to just draw S, then you have to draw V, and then you have to draw U. So this becomes the tertiary structure here. S, V and S, V and U. So this will be the structure that is belongs to the myoglobin here. And you must know that in this polypeptides N terminal is there. This is the N terminal of the polypeptide, and this will be the C terminal of the polypeptide. But this polypeptide will have a heme in the center. It will have a heme particle in the center. So this is the structure belongs to myoglobin. Now we'll see the bonds which are formed between different amino acids. Which bonds are formed there? We will see one by one. In this tertiary structure of myoglobin, first the interaction means the formation of bonds. The interaction between the non-polar R group. The first bond will form between the non-polar R groups. Second one, non-polar R groups means the hydrophobic amino acids and van der Waals forces. So the bond takes place between the non-polar R groups, that is hydrophobic amino acids and van der Waals forces. Now the second bond which will form, that will be the second, the bond forms or the interaction takes place between the bond formed between the charged R groups known as ionic bond. Means that the bond will form between the negatively charged amino acid and positively charged amino acid. Means between the acidic amino acids and basic amino acids. The bond will form between acidic amino acid and basic amino acid, known as ionic bond. It is called as ionic bond, right? Okay. So the interaction takes place between, or the bonds formed between the charged R groups, known as ionic bond. This bond will take place between the acidic amino acids and basic amino acid. Now the third bond which is formed, it will be the third bond, the bonds formed between polar chain called as hydrogen bond. So here the interaction between the polar chains will be hydrogen bond, or the bonds will form between the polar chains, that is called as hydrogen bond. And the fourth one is disulfide bond. Means the bond will take place between the SH group of cysteine So you must know that we have seen the SH group means that in insulin we have seen 2 polypeptides are reported by the Frederick Sangerhen. In the first polypeptide 21 amino acids are present, and in the second amino acid 30 amino acids are there. So total 51 amino acids are there in a insulin. So here you must know that the bond will take place known as SS bond, and this will take place between the SH group of cysteine from one polypeptide and the another polypeptide of the insulin. So you must know that the disulfide bond takes place here. So these bonds are necessary for the stability of the molecule of the protein or the structure of the protein. Otherwise, there will be no stability of the protein without these bonds. These bonds are helpful. They are meant to stabilize the structure of protein. So we have seen the tertiary structure here. Now we'll see the quaternary structure of the protein. So this will be the quaternary structure of the protein. Quaternary structure of protein. What will happen here? This is the structure belongs to hemoglobin. The tertiary structure of protein will again folded to the quaternary structure, and quaternary structure is said to be a polymeric structure. Polymeric structure means what? Polymeric structure means this structure of quaternary structure of protein will have more than 3 polypeptides. Therefore, it is said to be polymeric. If it will have one polypeptide, it is said to be monomeric. If it will have 2 polypeptide, it is said to be dimeric. If it will have 3 polypeptide, it is known as trimeric. And this is polymeric structure, means it will have 4 polypeptide in its structure. Hemoglobin will contain 4 polypeptides, and they are folded further from the tertiary structure. It will contain alpha 1. See, this is the structure belongs to hemoglobin. This is the subunit known as first subunit, second subunit, third subunit, and fourth subunit. This is third one, and this is fourth one. So here you must know that you should know that alpha 1 and alpha 2, they are just joined with the bond known as hydrogen bond. And alpha beta 1 and beta 2, they are also joined with the bond known as hydrogen bond. So here, if you see the structure of hemoglobin, 4 different units are there, and in the center, heme molecule is there. Heme particle is there, called as, in the center, it is heme. And if you see the alpha 1, alpha 2, it is not necessary that this alpha 1 and alpha 2 should be present in a same line. Alpha 1, alpha 2, beta 1, beta 2 can be also there, means they are present in such a manner. But here you must know that 4 subunits of hemoglobin is there, which contains heme. And the structure of this hemoglobin, which is stabilized, they are stabilized. Just now I explained you in the tertiary structure. It is the interaction takes place between non-polar R groups. Second, the interaction takes place between acidic and basic amino acids, called as Ionic Bond. So the interaction, the formation of bond takes place between the 4 different polypeptides to make the structure stable. Therefore, you must know that these are the bonds which takes place. The third one is hydrogen bonds I have explained here. The third bond which is formed by the hydrogen bond, or between the two different polypeptide, the bond will form, and the disulfide bond. Fourth one is disulfide. Between SH group of cysteine. I just told you that. I just explained you the structure of insulin. So here, what you must know, the disulfide bond will take place between the SH group of cysteine. The two polypeptides, they are connected with the disulfide bond. So these bonds, they make the structure, quaternary structure, stable. Therefore, we can see that these bonds are important to make the protein quaternary structure in stable condition. So friends, this is the answer for you. If a short note is asked in the examination, write short note on tertiary structure and the quaternary structure. So it becomes necessary to read books also, and which books you have to read, that I will take in another lecture. But you must know, for examination, for MCQ, reading of the book is very important, and to understand the full answer means that from full answer, this is the full answer, no doubt at all, but you have to read the books for the MCQs which can appear in the examination. So this becomes the direct answer for you which I have given. So if you like my lecture, comment, share it and subscribe it. Thank you very much. Have a good day.